Immunological techniques and applications. This is a continuation of the introduction to immunotechniques. And immunological techniques are based on antigen-antibody reactions. And these techniques can be used for the following uses. One, they can be used to detect an immune response, yeah, or maybe qualify or do a qualitative test or quantify the immune parameters such as the amount of antibodies. We can also use them to diagnose infections, and we can use where you do immunosurveillance within a population to know what antibodies do they have within their serum in a particular population. Now, some of the immune parameters that you can detect using a technique is the antibodies within the circulation that is in serum. What are the antigens either in circulation or those that are bound in tissues? We have cytokines, which are produced throughout immune responses. We also have sensitized B cells and T cells to measure for B and T cell activities, and also other immune cells such as monocytes, macrophages, and natural killer cells. So the classification of antigen-antibody reactions will be, we can have primary serological tests that use markers such as enzyme-linked immunosorbent assay, which is called ELISA. We also have immunofluorescent tests like flow cytometry, which we have done before, and we also have radioimmunoassay. So we did radioisotopes, so here we just have the utilization of radioactivity in the context of antigen-antibody reaction. We also have secondary serological tests like agglutination tests, complement fixation tests, precipitation tests, serum neutralization tests, toxin, antitoxin tests, and we have tertiary tests such as determination of the protective value of antiserum within an animal model, immune blotting, which we look at in the next part, and molecular techniques that are used in diagnosis. So we look at some of the tests that are utilizing antigen antibody reactions. One of them is precipitation reaction. So here, our antigen combines with an antibody in the presence of an electrolyte such as sodium chloride at a suitable temperature and pH. So it is antibody antigen in the presence of uh, sodium chloride. So the antigen and antibody complex forms an insoluble precipitate that we can now see or visualize, that is suspended in flocules. So the reaction can take place in liquid medium. It can also take place in a gel or agar or agarose or also polyacrylamide. So the principle of precipitation reactions is that we have a soluble antigen combining with an antibody, giving us a visible precipitate. We also have what we call lattice formation, where antigen binds with um, there are two antibody binding sites of two antibodies so that they form like a mesh. And we do have some examples of precipitation tests: single diffusion, or what we call radioimmunodiffusion, double diffusion, and then we also have immunoelectrophoresis. So this is what you are saying, that we have also the presence of formation of lattice. So for example, if you're having a polyclonal antiserum, so these antigens here, the yellow ones, will have different epitopes that are binding to different antibodies, and that leads us to the formation of a lattice. So we won't have any uh, precipitate being formed if an antigen contains only one single copy of the epitope. So if we only have single, you can see that the antibodies have bound, but they're not coming together to form a precipitate. So we normally have what we call a precipitation curve or precipitin curve, because now when we start this antigen antibody reaction, we can be observing the precipitate formation. So when we are beginning, we have an antibody excess. Yeah, we have a lot of antibodies. So when we have a lot of antibodies, this zone will not be a well-formed what we call a precipitate. Then we have what we call antigen excess, where we're having too much of antigen so that the antibodies are not able to bind. So we don't also have the formation of a lattice or precipitate. But we do have an equivalence zone where we have enough antibodies against enough antigens, and once they combine, they aggregate together and form a lattice that now forms what we call a precipitate. We can be able to see a precipitate. We look at a single immunodiffusion, a type of precipitation reaction. So what happens is that within this setup, your antigen is within the well, then you add your antibody. So one reactant is what moves, yeah. So once you have incorporated your antibody, then you have the antigen moving outwards or diffusing outwards. So as it is diffusing, it will come across the antibodies, yeah. So that if then they are specific, we will have this region where we are having excess of antigen, yeah. Then it will continue to diffuse outside, getting into more antibodies, and we will have an equivalence point around here, and that is where we will see a ring. Precipitate forms a ring that we can visualize. We can also do this when you're having different dilutions of the antigen, so that your antigen is still in the well, but you have different concentrations. So this, after you have added your antibody, then you can see the formation of precipitin rings here, depending on the concentration of your antigen. So as the antigen diffuses into the gel, it reacts with the antibody, and when the equivalence point is reached, that's when we have the formation of a ring. Yeah, and now the diameter in this case, because these are different concentrations, the diameter of this ring is going to indicate the concentration of the antigen. It is directly proportional to the concentration of the antigen. So we expect that this is more concentrated as compared to this one. We can also vary this one by now having the reverse, where we're having different antigens and antibodies now within the wells. The other time we were putting antigen, so we can add different antibodies. So we have IgG, IgG, IgA, IgD, IgE, and so on. And now we can determine how much level or quantity is there within the particular sample. We have what we call double diffusion or octalony, which is similar to what we have seen before. So what happens is that now you have your like you're doing it in twos or multiple. So the virus antigen is still placed within the well, but a central well in this case, so that now here you have your viral antigen, for example. Then these wells you put in samples, yeah, so that you want to see maybe these serum samples, do they contain antibodies against your viral antigen. So in this case, A and C are positive. They have reacted and formed a precipitin ring. Then B is negative. There is no formation of a ring. We also have what we call immunoelectrophoresis, which also is based on the formation of a precipitate. So now sometimes we don't just have one antigen within your sample. Sometimes you have multiple, what we call a complex mixture. Let's say you have just taken a sample of blood. It will have mixture of antigens from albumin or proteins within the blood. We have hemoglobin moieties. We have maybe cytokines, among others. We have some hormones, etc. So when you have a complex mixture of antigens, you can now still be able to visualize the many different antigens. But what you do first is first separate them. Yeah. So you punch out a gel, and then you add the sample which contains many antigens. Then you electrophorese so that you allow the antigens to be separated according to their size and also their charge. So after the electrophoresis has been completed, then a trough is going to be cut, or like a trench is cut in the gel, and antibodies now will be added. In this case also, the antibody mixture contains a lot of antibodies that can target different things within your serum. So as the antibodies diffuse into the agar, then we have precipitin rings being formed. Of course, when we reach equivalence zone, once we have the antigen antibody reaction. So this is what happens. So you have been able to separate your different antigens within your agar, right? Then you dig a trough within your gel, like this one, add antibodies. Once you have added antibodies, the antibodies are going to now go and start locating where your antigens are. So when we have the formation of rings, then you know that we have arrived the equivalence point, like here, the equivalence zones. And now you can be able to also determine the concentration by the diameter. And now this is how it looks as an image. So you can see the precipitate ring that has been formed. In this case, this is negative. You can see this is a positive reaction. So this test can be used to analyze components of your serum, and where you now place the serum in the well and then the antibody to the whole serum in that trough. And then you can compare also normal serum and now the patient serum. That way you can also be able to detect deficiencies. Maybe this serum is lacking a certain component that was not detected, and it was detected within the normal, and maybe it's lacking within your patient. So you can be able to also keep comparing. Now, those are precipitation tests. We have agglutination tests. Now, precipitation tests, we were coming from soluble antigen plus antibody forming an insoluble precipitate. But now we do also have antigens that are in particulate form, or they are whole cells, or they are already insoluble by themselves. So are we able to detect them? Yeah. For example, when you talk about red blood cells, yeah, how are we able to determine, let's say, blood groups? Because a red blood cell is a particulate entity or antigen. So this is now detected using this technique called agglutination test, which now resorts to the clumping of the antigen. So if they are red blood cells, they can clump together when it's a positive reaction. So the general term agglutinin is used to describe antibodies that will agglutinate particulate antigens. So if the antigen that you're dealing with is an erythrocyte or a red blood cell, we call this process hemagglutination. So all antibodies can theoretically agglutinate particulate antigens, but IgM, because it's high valence, we saw that it is pentavalent, is a good agglutinant. So this is one example when we do this test qualitatively, just to see if there is a presence of certain particulate antigens. And it is a common blood grouping test, where we have, for example, we have antibody A. You know that we have blood group A, blood group B, blood group AB, and blood group O. Then we also have the rhesus factor. Either you have the rhesus or you don't have. So you're rhesus positive or rhesus negative. So what happens is that um, you will now have anti-A, yeah, you add it to the patient's serum and you observe. Anti-B also, then anti-D. So for example, in this case, this patient agglutinated with anti-A, meaning that the blood had the antigen A on the surface of red blood cells. That's why there was agglutination. They did not react with anti-B, and it reacted with anti-D. So this person is blood group A positive. Now we can also use this test now quantitatively, and anytime we have quantitation, just like we did in precipitation, we had dilutions of your sample. So even here you can dilute or do what we call dilution, several dilutions like this one. So you have different patients, then you do some dilutions of each of the sample. Let's say 1 is to 2 to 1 is to 10 or 24. So you do dilutions, and Once you do dilutions, then you react your antigens with the antibodies and observe how agglutination occurs. So this is a positive control, this is a negative control. So you check for the dilution where we are having agglutination, the last dilution. So for example, in this case, this is 1:64. Yeah. So what we say is that the maximum dilution gives us a visible agglutination and we call it the titer. Yeah. So the results are reported as a reciprocal of the maximal dilution that gives the visible. So this is the maximum dilution that gives a visible agglutination, and you can see from here we don't have. So the titer is the reciprocal of 1:64, which is 64, and so on. So this is 8, this is 512. Here we don't have, so it's below 2, which was, this is the maximum, and we don't have, so it is below 2. Then we have 32. Now this one, we'll call it 128, but still we have a few challenges. So you can see like here we don't have any reaction. So this is a test that should also be repeated. Here we have 32 and here we have 4. Now we talked about precipitation using soluble. We have talked about agglutination using particulate, but it's possible now to check for a soluble antigen, yeah, by first binding it to a particulate antigen so that you achieve agglutination. And this is what we call passive hemagglutination. So remember, a red blood cell is a particulate antigen, but now you can add to it the soluble antigen that you want to detect, or you coat. Yeah, maybe it's a viral antigen, it's a polysaccharide, etc. So you coat and use these coated red blood cells to now agglutinate with antibodies, and this is now what we call passive hemagglutination. And it can help us to detect antibodies that are quite minute, antigens that are very small, but they are soluble. So it is very important, such as viral antigens. Now, we talked about radioimmunoassay, which we said it is used to detect antibodies and also quantitate now how much antigen was in that particular analyte using radioactivity. So a radioimmunoassay involves the separation of a protein from a mixture using specificity of the antibody antigen binding. That's why it's an radioimmunoassay, and it was introduced by Bassen and Yalo in 1960s, and it has shown like the first time they used it is when they measured hormone levels within the blood. So this technique can be used to detect micro quantities of protein, even viral antigens, antibody, structural proteins, vitamins, drugs, and also their metabolites. And it can do this in picograms, very minute samples. And that's why it is very usable in biochemistry and also in biotechnology. So how does it work? So I'm going to use this as the explanation. So what we do is we first have a radioactive antigen that is similar to your analyte. We combine it with the antibody, what we call the primary or the first antibody. So we expect that it has bound, we wash. Then now we add in our unlabeled. So it's kind of a competitive binding principle. Yeah. So we add now our antigen of choice, which is not labeled to any radioactive material. So we expect that it is going to compete for the already bound sites and displace some of the radioactive antigens. Yeah. Once it has displaced them, then we will add another antibody, which is now a secondary antibody, which is supposed to be binding to this primary or first antibodies. Yeah. So in the end, we have two. We have a supernatant, and we also have the precipitate, because this will cause precipitation. So this precipitate has bound to the antibodies, which we expect to be bound to uh, both the labeled antigen and the unlabeled. Now, what is important in this case is the concentration of bound versus the unbound. Yeah. So what happens is, if you take, for example, this here, this a secondary antibody that has bound to the first antibody. So if we were to measure radioactivity in this case, remember that the radioactivity is based on the red, which is a radioactive antigen here. So if we measure radioactivity here, in this case, you'll find that it will be low, because in this one, we have 3 of the blue and 1 of the red. So radioactivity here will be low. But being low then means that our analyte, which is the blue, which is unlabeled, was more. So it's an inverse kind of relationship. And that is why in this graph here, we will have, when we were starting, our red antigen, which is radioactive, was bound. Yeah. So it will start from high, then it will go reducing. Yeah. And we are expecting that it is reducing because we are increasing the unlabeled antigen binding. Yeah. So that means that we can inversely be able to get the concentration of our antigen, which is unknown, our unlabeled antigen. So we're able to use this graph and we can generate the concentration of our unlabeled. This is a very high specific test. Remember when you talked about cross-reactivity? So it's very highly specific because already the antibodies had been bound to another radioactive antigen. Then they were able to replace them. That means there's a lot of high specificity. Then high sensitivity because it's able to bind to very little or few picograms of your antigen, and it is highly accurate and precise. So the disadvantages, of course, radioactivity is a radiation. There are radiation hazards. Also requires trained personnel. Also, the labs have to be radioactive licensed, and they require arrangements. A lot of requisition of these radioactive materials, quite a process. Disposal, I think we talked about that at some point. So it may not be very used in our routine laboratories, but it is a very high specific test. Yes, I think now we have just one more.